Morning, everybody. We'd like to get started. Let's see, how do I do this? As is usual, I'm going to make a few color commentary comments and then introduce the Irina, who's going to introduce our speaker, although I'm going to make one little observation before Irina gets the opportunity to do that. Next week, our Grand Rounds is our town hall, which is going to focus on climate in the department, and we have a guest speaker for the beginning of that, who is Aisha Holder from Columbia University, who's going to be talking about microaggressions, and then we'll have a more general discussion about how people interact in the department. We are setting this up so that people can send in confidential questions that you would like to have discussed in the department. And if you don't know how to find that login site, just send me an email and I will find out what the website is to send your confidential comments or emails about microaggressions or other aspects of the climate in the department. So I'm not introducing this morning's Grand Round speakers, but Etienne Sibille, but I do want to make one little point which I found fascinating. There's something called neurotree, which everybody, as you establish your career, has. In other words, you don't make it up; they make it up for you, and it cites your intellectual grandparents and great-grandparents. If you don't believe in extraterrestrial powers, I am now demonstrating them for you. Maybe it's my phone. Can we do something to make that go? Okay, good, thanks. All right, good. So here you have Etienne Sibille, our Grand Round speaker. But let's trace his intellectual roots. Renee Henn, his postdoctoral advisor, famous neuroscientist at Columbia. And his advisor, Richard Axel, Nobel Prize-winning neuroscientist at Columbia. Then his intellectual grandfather, Linus Pauling. And then his intellectual fathers, Erwin Schrödinger and Niels Bohr. So this is who's lecturing to us. Great intellectual grandchild of Niels Bohr and Erwin Schrödinger. Not too bad. So, yeah, I'm happy to. So, yeah, that's right. That's right. A cat's good. All right. So anyway, so I thought that was really kind of interesting and neat. And but as I said, that's not the introduction of our speaker. Just a color commentary on the introduction. So in order to do the introduction, we'll bring Irina Esterlis up to introduce our speaker today.
I think that introduction is a lot more fun than mine. So as you saw before, Dr. Sibille trained with Renee Henn at Columbia University, where he became assistant professor and also at the New York State Psychiatric Institute. He was recruited to Pittsburgh in 2004 and became tenure faculty there. And then now he's at CAMH. He's the chair of the Campbell Institute there and professor at University of Toronto. He is a molecular neuropharmacologist with 20 years of expertise and over 120 publications in the field in human postmortem investigations and rodent modeling of neuropsychiatric disorders with primary focus on depression and aging. The goals or visions of his research program are to investigate molecular and cellular mechanisms underlying major depressive disorder, lifelong maturation or aging of the brain and their respective interactions, and to use this knowledge to develop strategies for novel therapeutic approaches. His group performs large-scale genomic, molecular, and biostatistical investigations of the pathology of these conditions in human postmortem brain cells. They also use relevant genetic and environmental mouse models to investigate causal links between identified pathologies, and they work with medicinal chemists, pharmacologists, and external partners to develop and test novel compounds for therapeutic efficacy. Today, Dr. Sibille will talk about his most advanced project, which is targeting alpha-5 containing GABA-A receptors in depression and age-relating disorders. And I'm very happy that he agreed to come so that we can learn more about his work. Thank you.
Thank you, Anna. So, thank you. I think this microphone works. Can you hear me? Okay.
Oh, did you turn it on?
I don't. Is it on? No, it's on now. Is that better? And I guess we need to turn this on. So thank you, John and Irina, for those kind introduction. I learned a lot about myself. I feel almost taller with such an ancestry there. So this is. But in addition to that, what I thought you were going to mention is my connection to Yale, because even I've never been. I have many collaborators here. Or colleagues at Yale, but being at Pittsburgh for 10 years and now at CAMH, there are many members of the Yale diaspora that have been my neighbors and colleagues. So I've always feel like somehow be a distant cousin of you guys, So, so what we set up, as mentioned, so I'm a molecular neurobiologist by training, and we've over time I've been interested in a cellular molecular basis of depression, but as you'll see from the beginning, we always used the effect of age as an internal control for our studies, and the idea was if we can't see the effect of age, forget about the effect of depression or something like that. But over the years, it became really intrinsic. Those two lines of research have become intrinsically linked because we see this at the molecular level. We see this evidence of advanced or accelerated aging in the context, not just depression, but schizophrenia, bipolar, and even extending to neurological disorders such as early Alzheimer. So I'm not going to focus on aging now, even though I'm going to mention it quite often. And you'll see in our drug development project, the implication are for cognitive dysfunction in depression, but in late life as well. And I think in the context of other with Irina, I think you're setting up some symposiums where we can talk more about aging in particular. So, GABAergic deficit in depression. In terms of disclosure, just want to say that I'm a co-inventor on a patent application describing the compound that I'm going to talk to later. So I was reminded to let you know that not everything is perfect with those compounds. There are many caveats as well, but I'm somewhat biased. So, getting to biology, I'm going to jump directly into the focus of our investigation. So within the cortex, in the brain, within cortical structure, there's a canonical organization of a few cell types that are responsible for, if you want, processing information. And this cortical microcircuit is also responsible for balancing, if you want, excitatory activity with inhibitory activity. And which is, so in short, maybe I can use this one. Pyramidal cells are the major, of course, excitatory neuron. They propagate information, but they're under the control of different cell type, GABAergic cell type, which are inhibitory by nature. But those different cell type can be identified by, let's say, the neuropeptide that they express, so somatostatin, VIP, or PV, by the electrophysiological property, but also by the cellular compartment that they target. So parvalbumin tend to target the perisomatic region and therefore responsible for regulating output of information. Somatostatin, which I'm going to talk quite a lot about, actually regulates information input or integration of information on the dendritic tree. And the vasoactive intestinal peptide, or VIP, regulate somatostatin, so they involve in disinhibition. If you want, they inhibit the inhibition, therefore allowing performing disinhibition or allowing information to go through the pyramidal cell. So this is a canonical simplified microcircuit, and it is known that the excitation-inhibition balance is really maintained by this microcircuit at the millisecond level. So electrophysiologically, that has been shown. There's a really nice optogenetic study that have confirmed the details of this regulation. But in the context of lifelong or chronic situations such as aging and neuropsychiatric disorders, it is thought that the homeostasis within the system is maintained by long-term adaptation regulated by altered pattern of gene expression. So so it becomes more efficient if you want to change the structure of your neuron to maybe make them more reactive, less reactive, more neuron, less neuron. So structurally, this long-term homeostasis is established and can therefore be, what happened there? I think I pushed, okay. And can therefore be, if you're on reverse engineering by identifying pattern of gene expression under those conditions. I'm just going to show you one example related to somatostatin. So a few years ago, we performed gene expression analysis in the human postmortem brain, and we showed here, for example, that some markers of those cells, such as somatostatin, was downregulated in depression compared to controls. Another marker, calretinine, but the same works for VIP or PV, were not affected. But there's also a lot of variability. If you can see the intra-subject variability. And we've shown that up to 30% of these variants can be explained by the normal effect of age. And by that I mean, even in normal subject, in the black dots, you can see that between the age of 20 and 60, you will lose close to 50% of the expression of that gene. And you can see in depression, there's some noise, but it seems to occur on an earlier trajectory. So regardless of age, depression subjects tend to have low expression of somatostatin. So you have this parallel or potentially anticipated, some people talk about accelerated aging that we can actually see at the molecular level. And so over the years, we performed multiple studies. This is a meta-analysis of a large-scale expression study showing that, on average, this is a replicated finding that somatostatin is downregulated across, in both male, female, and across different regions of the prefrontal cortex as well as amygdala. And again, no change, no effect on PV-VIP. So there seems to be a selectivity occurring here on those markers. And similarly, there's an effect of age that parallels the effect of depression. I'm just showing you here, just a log ratio, so of changes. You can see in depression, it seems to have an effect that's always of greater magnitude and greater significance that what we see in the context of aging, specifically affecting those neurons. I'm showing somatostatin, but cortistatin and NPY are two neuropeptides that co-express to some extent with somatostatin. So this whole module seems to be affected, and overall there's no effect on those two cell types, suggesting a selective vulnerability, selective downregulation of the compartment that regulate information input in the context of age and accelerating depression. We perform a few additional studies to know whether, is this a reduction of cell number? Is this a reduction per cell? I can use that using, in situ hybridization, and we've shown that in the context of depression, it is a reduction across all layers, because the question is always, is it specific to one layer? Those different cells have slightly different function, and we can learn a lot about potential underlying mechanism. And we've shown that this downregulation affects SST neuron across all layers in male and female. And when you look at cell number, there's actually a reduction per cell rather than missing cells. So those cells seem to be losing their phenotype, their function, which from therapeutic purpose is actually interesting because it means the cells are still there. Maybe we can bring them back to life if needed. In the context of aging, the situation is a little bit different. So again, this is prefrontal cortex in human subject, using RNA scope, fluorescence in situ hybridization this time. And we've shown in this case that there's actually a reduction in detectable cells in the absence of total changes, number of total cell number. So it seems like when the situation, either the different mechanism or when the situation becomes chronic over decades, there may be a loss of cells. And so some slight differences, parallel trajectory, but slight differences in potentially in the phenotype and potentially underlying mechanism. A few years ago, we did a survey of the literature, and overall, this is actually a very well replicated finding. Reduce somatostatin depression, not just in depression, but across psychiatric disorders. So here we have listed Parkinson, Alzheimer, major depression, schizophrenia, bipolar, and you can see that in all cases across different brain regions, there's really good, robust, replicated evidence of decreased expression, either at the RNA or the protein level, of this marker. So well-validated, replicated pathology, and which also occur in the context. So somatostatin, if you want, we use it in this case, and we've shown that also as a marker of the function of those GABAergic neurons. And just to make a long story short, you can basically equate SST function with the function, the neuropeptide function with the function of the GABAergic cell. Because when we affect one system, the other system changes in parallel. So the point is that those changes in marker occur in the context of a lot of information that suggests that, or that show that there is reduced GABA level, GABA function in the context of depression, in the context of CSF, the work of Jerry Sanacora using MRS showing low GABA tissue level, reduced marker of GABAergic neuron has been shown by us, but by many other groups in the context of depression. You can use TMS, transcranial magnetic stimulation, to actually assess cortical inhibition, and that has been shown to be reduced in depression. In terms of function imaging, Georg Northoff and others have shown that that GABA contribute to some of the other phenotypes that I see in the context of depression, such as changes in resting state activity. And Vilma Gabay have confirmed, if you want, those decreased GABA level in the context of depression and really correlated with the anhedonic component of depression, actually. So just to provide a little bit of a background on the robustness of this phenotype, if you want. So based on that, we a few years ago, we decided to focus and investigate more potential mechanism and therapeutic potential, and potential for therapeutic targeting. So one of the early hint in terms of mechanism came actually from the human postmortem studies. So what I'm showing you here is that the profile of downregulated, so blue means downregulated, those genes are markers of synaptic markers. They're all downregulated, in this case, in the amygdala, in human subject, confirmed by PCR. But what was striking is that we remembered from a previous study that this is very reminiscent to pattern that we see in mice when you reduce BDNF expression. So we all know, that neurotrophic function is downregulated in the context of this disease, BDNF. But for us, we see so we have this pattern in human, and you can see here using transgenic mice that either reduce the constitutive activity of BDNF. BDNF has a circadian oscillation, so in het mice you have this lower level, or in mice that reduce the activity-dependent release of BDNF. If you look in the frontal cortex of those mice, you can see that between these two manipulations, you can recreate the pattern of dysregulation that we see in the human. So this was pretty good evidence, if you want indirect evidence, that the pattern we see in human may be due to reduced BDNF function. And interestingly also there's a role for aging. We looked at again, it is known, so this is a confirmation of reduced BDNF, so more direct information, both at the RNA and the protein level in the context of depression. But again, the effect of age, it is known that BDNF goes down in normal subject with age. Again, between 20 and 60, you will lose 50% of expression of BDNF in normal control. And again, as I've shown for somatostatin, it seems to be on an anticipated trajectory in the context of depression. So again, this is noisy data, but you can see that overall, regardless of age, subjects with depression have lower expression of BDNF compared to age-matched control. And we went further on. There are many genes that depend on BDNF, so you can use either bioinformatic or evidence in the literature to identify a core of genes that are known to be BDNF dependent. And when you look at the trajectory of those genes in control subjects, so each point is the summary of age-related changes for, let's say, a set of BDNF dependent genes that either go up or down. And you can see in control in blue, you have this upward or downward age-related trajectory. And again, in depression, in this case, you see a clear acceleration pattern that was significant in the slope. So we know that BDNF is an important upstream factor, but this tells us that there are additional factors that may interact with reduced neurotrophic function to accelerate this pattern in the context of depression, and with this idea that the pathology gets worse over time, you have some potentially leading to treatment-resistant recurrent depression. So this may be a potential, correlate at the molecular level. But BDNF is this very broad neurotrophic factor that affect many functions. So how can we potentially learn a little more in terms of mechanism? So for this, we went, as you know, BDNF is a complex gene that has multiple exons that through alternate splicing can form multiple variants. And so we looked in the human postmortem at the expression of all those variants, and we also know that those variants target the transcript, the BDNF transcript, to different cellular compartments, with some restricted to a cell body and some that extend to the dendrites. And we look at that, the only one that was significantly downregulated was what's called a 3' untranslated region. This 3' UTR is known to target the transcript to the dendrites. And we looked also again, this is a expression change of, and this was in parallel to expression change in gene that correspond to markers of dendritic targeting interneuron, but not perisomatic. So again, the same pattern that we saw before, this selective effect on GABAergic neuron happening in dendritic targeting. But now information that maybe this molecular tag, if you want, that bring BDNF to the message to dendrites so they can be used in an activity-dependent manner would be specifically affected. And we show that actually, yes, the somatostatin is directly correlated with this transcript, none of the others, and genes that are, and this is for all genes that are affected in the context of depression related to the synaptic function. They're all correlated with this one, not with the other transcripts, etc. So pointed us to this regulatory function aspect of BDNF as a potential mechanism. So we went on to actually knock down the function of this specific transcript. You use an shRNA approach packaged in a viral vector, in an AAV vector. And we show in cultured neuron that if you knock out specifically the 3' UTR, you have a reduction in, you generate a reduction in dendritic length, dendritic complexity. So we show an effect on the phenotype of interest. So then we went into mice. If you inject that in prefrontal cortex region corresponding to the precingulate and prelimbic, and to make I have to put my glasses on there so we can see. So what we did, wanted to look at specifically downregulated the 3' UTR BDNF in the frontal cortex, and the question is it sufficient to induce morphological, anatomical, and behavioral changes associated with depression? And the short answer is yes. So morphological, as shown here on cell culture, in context of gene expression, we show that it takes 2 to 4 weeks to downregulate the 3' UTR. Eventually, you get downregulation of somatostatin, so confirming the dependency. At the behavior level, what you can do, the model we use to induce, if you want, depressive-like state is unpredictable chronic mild stress. So if you're not familiar with the models, it's a period over 4 to 5 weeks. You submit mice to a couple of stressors a day in an unpredictable manner, and it leads to a syndrome that includes increased anxiety, decreased interest for palatable food, induce neuroendocrine changes, usually elevated cortisol levels. So, different aspects that would look like a depression syndrome in mice if that were to exist. And you can see that in mice that have a reduced aspect of BDNF, this dendritic targeting, these mice are actually, you can see here, you can focus here. This is just a Z-scoring of different behavior. You just summarize a lot of information. You can see at baseline, mice with reduced BDNF are more emotional, so it increased anxiety, depressive-like behavior, both at baseline and after chronic stress. So altogether, this suggests that there's a causal link between that specific aspect of BDNF function and the molecular and behavioral phenotype of depression. So we are, I'm going to stop here on the BDNF because, based on all that, we know that, it occurs in humans, it's causal, and there's some specificity, so it's potentially targetable. It's a challenge to target neuroplasticity in terms of therapeutics, but we're working on that, based on this level of evidence. But if we step back again, go back to somatostatin in terms of causality, we've also shown that, just simply if you remove somatostatin from a mouse system, a knockout in the context of a just constitutive knockout, we can recapitulate behavior, molecular, endocrine phenotype of depression. So mouse lacking somatostatin, you can see here replicate decrease in GAD67, the other, as well as the other marker of dendritic targeting, as well as BDNF. They're all downregulated. So even though somatostatin is downstream from BDNF, if you knock out BDNF, you also affect, your feedback mechanism. So this is what I mean by, you can really use somatostatin as a proxy marker of the function of those cells. And again, in terms of behavior, you can just focus here if you want. That both before stress, mice lacking somatostatin are more anxious, depressive-like, and as well as after stress. And you can also see from the neuroendocrine point of view, it is known that somatostatin control hypothalamic release of the increase the HPA axis. So you do have an increase in basal corticosterol level. We've shown that this is not sufficient if you want to induce the overall phenotype, and as is well known that increased CORT is a model, and that's mostly due to the fact that those changes are at baseline. Stress-induced CORT level do not change, so there's just an increased baseline, maybe primes the system for increased vulnerability to stress, but it's not sufficient on its own. We also wanted to know, what about if we just knock out the function of cells of the somatostatin neuron in frontal cortex only? So for this, we use a pharmacogenetic, chemogenetic approach, the DREAD approach, which offers both temporal and regional specificity. So in an adult mouse, you can inject, if you want, this modified muscarinic receptor that will inhibit the function of those cells. So this is just some of the technical details, but basically the DREAD approach. And so we do that, do an injection, wait for 2-3 weeks, and then do some behavioral testing where we activate those DREAD receptor with the acute injection of clozapine N oxide, which is a ligand that is specific to the DREAD receptor that we introduce. So it doesn't have any other function. And you can see that acutely, and this results in basically inhibiting the function of the somatostatin neuron. So you have a normal, so in contrast to a knockout, which may have developmental compensation, this is an adult system, intact, and you acutely block the function. It's like optogenetics using a chemical approach. You can see that blocking the function increase anxiety in the elevated plasmase, in the open field, and to test for for anhedonia. Overall, again, if you z-score this behavior across tests, you have a very robust and significant increase in emotionality. Again, telling us that the function of those cells is critical for the behavioral phenotype. So just to summarize a little bit at this point what the characterization of this pathology, we show in the context of human that it's reduced across cohort, across region. We've shown that BDNF, stress, aging, I haven't shown you, but sex, all moderate. These are additional factors that moderate, potential causal factor that moderate this expression. And using mouse studies, we basically begin to identify potential mechanisms linked to neurotrophic function, linked to excitation inhibition balance, but importantly, we show that there's a causal link. So based on all that, we say, well, it occurs in human, it's causal, we have a mechanism, can we target this, and what are the potential mechanisms? So we summarize that in a paper recently published, that the idea is depression and stress. If we zoom into this synapse, which is really the dendrite, depression, stress, aging may lead to reduction of BDNF that is localized to those dendrites. This leads to a reduced supply of BDNF to those cells that are highly dependent on it. So reduce dendritic complexity, indirectly, reduce GABA function altogether, leading to altered information processing in this compartment and leading to mood dysregulation, and as I'm going to show you again, cognitive dysfunction, we believe, as well. So this is an overall model. In terms of, but there's indication that in addition to mood, there's potentially a critical role for this functional module in cognitive processes. And one of the assumptions there is that, in a healthy microcircuit, the function of those cells is really to inhibit pyramidal neuron. So if you look in layer 2, 3, it is known that pyramidal neuron output is very sparse, and this is directly related to the inhibition for this more active somatostatin neuron. When stimulus, when information comes in, in the context of external, internal stimulus, there's an inhibition of somatostatin neuron that occurs, actually through VIP neuron. But basically, the firing of somatostatin neuron decrease, and this allows, if you want to disinhibit pyramidal neuron, leading to much greater pyramidal neuron output. And he thought that this signal, to noise ratio in pyramidal neuron output is encoding information. So high signal to noise ratio is important for proper information processing. In the context of in a pathological context, we know that somatostatin neuron is down, the function of those neurons is downregulated, and this is expected to lead to less inhibition of pyramidal neurons. So if you want, your background noise is higher. Probably doesn't change when stimulus comes in, but what is changed is the signal to noise ratio. So this, high activity to higher baseline activity basically is speculated to lead to altered integrity and content of neural code. So the speculation is based on, there's a lot of optogenetics that confirm or that suggest this is a highly probable mechanism. A year or two ago, I decided to collaborate with John Murray from the department here in terms of developing computational modeling of this. So John can probably tell you much better than I can do, but using a relatively realistic biophysical model of the microcircuitry, he can, if you want, model working memory. That is the sustained firing of a population of neuron upon the administration of a cue. And typically when you bring a distractor, a different cue, what happens, a certain neuron notice this cue, start firing, but very quickly that disappears, and the information is the main information is maintained. Now you can model reduce inhibition, so reduce function or reduce number in this case of somatostatin neuron impinging onto those dendrites. And what you can see that the output is actually an increased distraction. The distractor cue actually takes over in terms of pyramidal cell function, and the primary memory is lost. And this is quantified here, distractibility, or I have to come here to read the distractor index if you want. So you have a decrease in the context of somatostatin. You can reverse this if you boost dendritic inhibition. Interestingly, if you boost inhibition on perisomatic through increased PV function, you further decrease, you further affect those working memories. So again, suggesting that there's a potential mechanism for a role of this downregulation of information processing in cognitive, potentially cognitive symptoms in disease. So based on all that, we thought this was enough information to try to focus on potential targeting for new drug development, if you want. And so we looked at what are the molecular components of those synapses, what are the potential strategy. And so this is a somatostatin neuron that targets the dendrite of a pyramidal neuron. It's by itself activated by excitatory and inhibited by GABA neuron. And the many potential site of action. And interestingly, the potential convergence with existing or emerging antidepressant modality. I think Ron Dumont's group showing that scopolamine, a fast-acting antidepressant, may be mediated by blocking the receptor on somatostatin neuron. The same may be occurring in part for ketamine. Benny Lucia, for example, has shown that if you genetically activate the function of somatostatin neuron, you have a very robust antidepressant effect. TMS is known to actually, and I'll go back to that at the end, it was speculated for a long time, it's been shown now to transcranial magnetic stimulation is known to activate excitatory afferent onto somatostatin neuron. So TMS may indirectly boost the function of those cells. So, I'm not saying this is all occurring through those cells, but there's a good convergence in mechanism suggesting a role both in the pathology but in the therapeutic modality also for this signaling pathway. So we decided to focus on those postsynaptic receptor in the GABA system. So because somatostatin are after all GABAergic neuron, and postsynaptic and perisynaptically opposed from somatostatin neuron, you have this specific GABA-A receptor that contain the alpha-5 subunit. And a few interesting features of this particular gene is that it's expressed specifically in dendrite. It's specifically restricted to cortical layers and to the hippocampus. I think it mediates through tonic inhibition. I should update that. There's a very nice paper that came out a couple of weeks ago that actually showed that it mediates both tonic and phasic inhibition. So it's both synaptic and extrasynaptic. And so the idea would be that if we were to use a positive allosteric modulator, if we were to boost the function of those cells, we may target the pathology and reduce symptoms, both potentially cognitive and mood symptoms. So that's just the overall, hypothesis for this therapeutic approach. The GABA-A receptor, I think as we know, it's a pentamer. It's a chloride channel that is formed by different subunits, alpha, beta, gamma, delta, etc. And this is a target of benzodiazepine. Benzodiazepine is actually a different site from the action of GABA, and it's known to modulate the function of GABA-A receptor that contains specific subunits. So actually alpha 1, 2, 3, and 5. And so it's great. And as we're discussing with Jerry yesterday, I was saying maybe benzodiazepine are the most reliable drugs in psychiatry. They will have an effect. And that may be also the problem in that there's been an overwhelming known anxiolytic effect and then sedative, but it has potentially masked the potential, the therapeutic potential for targeting cognition, for targeting depression as a whole. And this is due to the fact that benzodiazepine basically are not specific. They activate all GABA receptor with these 4 subunits. And it's been known for a while that now, you know, for the last maybe 15 to 20 years, genetic studies have shown that those different alpha-containing GABA receptor mediate very different function. They have different brain localization, different cellular localization. So you know, for example, alpha-1 mediates the sedative effect of benzo. Alpha-2 mediates the anxiety and anxiolytic effect. Alpha-3 was a little bit unknown until recently. Alpha-5, which is our target here, until recently was unknown, mostly because of dominant effect of the other subunit. But it's known now due to its localization, both anatomically, cellular, and function, that this regulation at that level through alpha-5 may be involved in cognitive function. So the idea is, you know, targeting specific alpha-5 GABA-A receptor may actually uncover novel therapeutic role for GABA potentiation. So based on that, we started to collaborate with a medicinal chemist, Jim Cook, University of Wisconsin, and the idea was to develop a compound with increased selectivity and affinity at the alpha-5. So we go, and the idea is we started with the imidazobenzodiazepine, which is a hybrid molecule that is known to bind our target. The advantage also is that it's been in the clinic for many years. These are safe molecules, so the probability of success would be greater. And you go through the typical medchem optimization where you have lead compound and then you improve, and you test, and you go through this feedback. And I'm going to show you data with one of the early compound, this alpha SHO53, which is a positive allosteric or alpha-5 PEM. We've shown that, you know, again, if we just focus on this bottom graph, you can show that after chronic stress, this summary value, z-score of emotionality, is significantly elevated. Interesting, after a single injection or chronic injection, you can block. So you basically have an acute and a sustained, so rapid, if we want, acute or chronic antidepressant effect. This looks great, but you know, not all data was this pretty. This actually worked in female only. Male, we had a much lower signal, and it wasn't as reproducible. So, but that was one of the parent compounds. So we went through all this medicinal chemistry effort over the last few years, where here's our parent compound, and we modified different residue, especially this upper right end of the molecules, which we know is responsible for the binding pocket into the alpha-5. And we've done multiple modifications, tested all those behaviorally, also looked at activity of this compound, if you want, for electrophysiological activation of our target of interest. We also looked first, of course, do those compounds go to the brain? What are the PK? What are the half-life? You know, other potential drugs. But in recombinant receptor, you can see here, what I'm showing here is the activation, the potentiation of chloride currents when you put GABA and this modulator together. For example, here you can see that alpha 1, 2, and 3 containing GABA receptor activated, alpha 5 much greater potentiation level. 4 and 6 are internal control. We do not expect any potentiation. And you can see that those compounds have different profile. This is relatively selective, you know, you're at certain dose, but this is pharmacology. You know, selectivity is just a function of dose. And we show that here at 100 or 1 micromolar. This compound, GL275, is very close to what a benzo is, actually, which is high potent activator of all 4 subunits. Benzo would be actually even higher and has higher affinity. But it gives us different tools if you want to test them behaviorally. And I'm just showing you some behavior with 3 of the lead compound for which I showed you the EFIS data. You can see in the elevated plasmase, so testing the anxiolytic effect of benzo, you can see that benzo, a robust effect, increased the time spent in the open, threatening compartment of this maze. And you can see all 3 compound dose-dependently have this nice relief of anxiety, anxiolytic effect. In the forced swim test, which is a test for predictive antidepressant activity, benzos are known not to have any effect. We confirm that, but you can see that all our three compound on your ligand dose-dependently have antidepressant potential. So this, we already have now a separation from classical benzo in terms of behavior profile. In terms of cognitive screen, we use a Y-maze. So this is a test for spatial working memory. A mouse is put in a maze. It is allowed to explore, one arm, and then you take it out. 30 seconds later or 90 seconds later, you put it back, and the mouse is very curious, and will just will learn to go to the other arm and vice versa, and you can do that successive tests with 90 seconds interval. The cognitive load increase. The point is that after a week, a mouse will perform at 90% accuracy. They learn this task quite well. It's relatively robust. And the idea is to see, can we disrupt this function and then reinstate it with our drug. So this is our assay. And I'm showing you here. So what we use is chronic restrained stress. We can, to have it here. You can do just a simple restrained stress for 1 week. And this is enough to basically bring down spatial working memory to almost chance level, from 90 to 50 or 60, and with 50 being just random. And you can see that the two compounds here, very nice dose response, reversal of this cognitive deficit. Again, acutely, this is 30 minutes after injection. And GL275 here, a little, same reversal. There's a little bit of sedation starting a higher dose. And again, it's known benzodiazepine have no effect. If any, they worsen cognitive function. So again, second and quite interesting dissociation of effect of behavioral profile. Until what time do we have?
11:30. See a half hour.
Oh, great. I have only maybe 7-8 slides, so we'll have plenty of time for. So this is in the model of, if you want, adult stress-induced cognitive deficit. And as you've seen, from our previous slide, we're very much interested in trajectory with age. So what about cognitive deficit in the context of normal aging? So here we take mice that are 24 months old compared to 3 to 6 months old. And you can see that young mice perform as before, 90%. Old mice, basically, without any intervention, have very severe deficit in spatial working memory. And you can see that here, after a single injection, 30 minutes later, your old mice are able now to remember the training over the past week and perform actually almost, they're not significantly different from control, from young control. And this is after, so this is one compound, this is a second compound. And again, benzodiazepine have no effect in those tests. We've shown that not just acutely, but a big question with the benzo, we know it induces desensitization and the effect wean off with repeated exposure. And here we show that after 10-day treatment, you still have this very robust procognitive effect in old mice. Actually, we went up to 8 weeks of daily treatment and show that it's still, the effect is still there. So there's no desensitization, no evidence for side effects. Again, after chronic exposure. So we, based on all that, we actually, in the process of trying to move those compounds closer to the clinic. What I'm not showing you is, we have all those background information on this compound. We've tested again, their proper pharmacokinetics, toxicity. They do all cytotoxicity, etc. So we have a lead compound. So based on this premise that reduced GABA cell function occurs in the context of depression, other brain disorder, and during aging, that augmenting GABA SST cell function, just 1 second. So augmenting the function of this pathway by targeting The postsynaptic receptor, the alpha-5 GABA receptor, has pro-cognitive effect in both stress and aging models. Those novel molecules have drug-like property. So we put a team together. We've been working in the past 2 to 3 years, including tech transfer. We have IP on those, VC. So we're in the process, if you want, to get to the next level, which is really not what we traditionally do in academic settings, which is harnessing the expertise of biotech and pharma to process this compound through the drug development for, leading to a phase 1 study, hopefully in a couple of years. So we got support, we have interested partners, and when we got grants. And the idea, if you're on the indication, would be cognitive dysfunction. I've showed you only in terms of memory and spatial memory, working memory and spatial working memory, but we also performing multiple studies for additional domain regarding to prefrontal cortex function, cognitive flexibility, etc. But we know that cognitive dysfunction, if you want, are prevalent in depression, in other psychiatric disorders. They occur during aging. They actually define mild cognitive impairment, and they're very critical in Alzheimer's disease. They're also associated with increased suicidality across disorders. And yet, there's no drugs, targeting cognitive dysfunction. The cholinesterase inhibitors, if I understand it well, even in Europe, they stopped reimbursing them. They said, forget about it. They don't work. Let's not give them. Let's not waste everybody. Let's not create false hope. So there's an incredible unmet need. So this, hopefully, is in addition to the fact that those compounds have potential antidepressant, anxiolytic, and procognitive effect. And what I didn't show you is actually depending on the profile. So this is where those compounds are not perfect. Selectivity in the context of GABA A is almost impossible, to be selective for an alpha 1, 2, 3, or 5. And actually, some of the compounds have great selectivity for the alpha 5. But when it gets to a high selectivity, we actually lose the effect. So lower affinity and efficacy at the alpha 5 is important, but we also think we need actually a tiny bit of alpha 1 and alpha 2, and we confirm that by blocking those separately. So the exact formula, if you want, for activity of this receptor may correspond to the fact that one regulates dendritic inhibition, information input, the other regulates information output, that there may be a rebalancing that is needed across the compartment for optimal therapeutic output. So we have some ideas. This is work in progress, but it's not predictable. The behavior is not fully predictable based on the EFEs and PK characteristics. So it's quite interesting problem right here. In terms of drug development, the idea also is that we may not want to treat anybody. We may want to think about a cross-diagnosis approach. Sometimes we talk a little bit this notion of this may be similar to a cholesterol effect for brain function. In cardiovascular disease, if you have high cholesterol, you're at risk for multiple disease. So here we would say, GABAergic dysfunction affecting this compartment are present across multiple disorders. They may or may not be responsible for the phenotype based on additional and potentially, competing or synergizing additional pathologies, such as, for example, PV disruption in schizophrenia. So there's complexity there, but the idea is that, same as cholesterol, cholesterol may be not directly responsible for the illnesses, but it's a valid target on its own. By targeting high cholesterol, you reduce the risk of multiple cardiovascular disease. So here, by targeting this GABA dysfunction, we believe that there's a potential for affecting symptom dimension across disorders. And for this, we actually need a stratification tool. We can't measure it the way we measure cholesterol in patients. But actually, transcranial magnetic stimulation is a very good non-invasive tool to measure degree of cortical inhibition. So if you're not familiar with the approach, so this, you apply a magnetic field to the surface of the skull, so it's non-invasive, and it's known to activate, to induce action potential, or to activate neurons in right below, in the superficial layer below. And you can do that, for example, you can do that over the motor cortex and look at activation, do simple electromyographs. So you can titrate to a threshold, and then and you can do, for example, the same in electrophysiology. We can do pair pulse. You can do pair pulse activation of the motor cortex, and you can look at the response in muscle. So it's very non-invasive, peripherally, relatively simple to do. It's known that here and it's also known that it's been shown now through a really nice optogenetic study that TMS actually, it's always been known to only just very marginally penetrate the cortical layers just because of the physics of the waves. So it's always been known that it would only affect superficial layers. And they've known that it's been shown now in this paper that TMS actually activate the glutamatergic projection onto GABA neuron in the superficial layer, and this happens to be the one that target dendrite. So actually, I just realized this morning, this is actually a wrong graph. This should be a projection, should be here. So it's indirect, it's relatively direct evidence in the mouse here that TMS activate the cell that we see to be deficient in the context of the illness and disease. So it may actually be a good marker for the pathology in human. And not only that, but when you do those, these measures are known to be deficient in the context of depression. So if you do this pair pulse experiment, in a control subject, if you do one pulse, it induces what's called a cortical silent period, a certain period of time during which GABAergic function is active. And it's been shown that this CSP, or this silent period, is reduced in the context of depression, whether it's medicated, unmedicated, euthymic, or treatment resistant. So it seems to be a trait phenotype in the context of depression, where this reduced period is indicative of reduced inhibition. So the idea there is that TMS, may index SST-related GABAergic pathology, and it can be used for patient stratification. So the idea is, in terms of personalized medicine, it would be patient with depression, potentially remitted depression, cognitive dysfunction, and reduced cortical inhibition. This is the targeted population that would benefit from alpha-5 treatment. And so my last slide, I just want to summarize that said, hopefully provided you information showing you that both aging and brain disorder impact the somatostatin positive GABAergic component of the cortical microcircuitry, and that this is relatively specific, and that targeting this signaling module has potential for reversing the pathology and for improving cognition, cognitive and mood symptoms across brain disorder, as well as during aging. And with this, I'd like just to thank the group from my lab, essential collaborator, as well as a funding organization. So thank you very much.